Hello, my name is Jari Forsström and I'm a specialist in internal medicine. I will shortly tell you about pharmacogenomics and its potential in clinical practice. Genomics is used in different fields in medicine and also in hobby, but the pharmacogenomics is the field of genomics that is closest to clinical applications and also can bring most benefits and cost savings to healthcare. And I'm now concentrating on pharmacogenomics. Why it's a good time just now? The cost per genome has gone down rapidly, and now it's really possible to use genomic testing in daily clinical practice to help patients in medication. We have known for decades that the same dose is not fit for everybody, and some get complications with a drug with the same dose that others get good benefit, and some even may not get a benefit at all. So there's a significant costs and harm for patients, and with personalized medicine we can reduce quite a lot of these problems. What is the size of this problem? If we calculate the number of human drugs that is used, about 7% are those where the response varies a lot among patients. So with genetic testing, we can better adjust the drugs for the patient. But if we calculate the number of prescriptions, it's about 18% of prescriptions, meaning that they are very common drugs, and 1 out of 5 prescriptions are those where pharmacogenetic testing would be relevant and helpful for the patient. If we think preemptive pharmacogenetic testing, the most relevant genes for this kind of testing would be the genes listed here, SUB2D6 to C19. I will talk a little bit more about those genes, but skip the others today. But it's a one-time investment for the safer medication for the whole life for the patient. So even if the cost would be 200 to 300 euros, it's only one-time cost and the results are permanent. So it's good investment for safer medication. Then I give you a short example on SUB2D6. This was a very, very traditional example of codeine and the importance of SUB2D6. Codeine is a prodrug that is metabolized to morphine, that is the strong painkiller, through SUB2D6. Earlier it was just mentioned that about 10% of codeine is transformed to morphine, but today we know better that there's a lot of genetic variation in this. Some patients who are poor metabolizers of SUB2D6 may produce only 1% or less morphine from codeine, and that's why codeine is not working as a painkiller for those patients at all. But some are ultra rapid metabolizers, and they can produce very large amount of morphine with SUB2D6 enzyme from codeine. Even 50% of codeine is transformed to morphine, and they can get morphine intoxication. And there has been some cases where they had died with morphine intoxication. And that's why it raised a big interest in SUB2D6. The new findings on SUB2D6 led new recommendations to codeine dosing. Today, codeine is not given to children below 12 years, and it's not given to patients who are known to be ultra-rapid metabolizers because they are at the risk of intoxication. However, this test is not routinely done for each patient before codeine is given, but it will change quite soon. Another slide describes the importance of SUB2D6 in psychiatric drugs. There are a lot of different drugs, and in these columns you can see the different clearance of the patients in different groups. If the patient is ultra-rapid metabolizer or poor metabolizer, the difference may be tenfold, for example, desipramine. So everybody can understand the same dose can't be the good dose for everybody. CYP2D6 is very important gene in drug metabolism, and there are also new drugs coming to the market. This is one example, quite new drug, Serdelga, and here the authorities have already asked the pharmaceutical company to test the drug for different phenotypes. So there's dosing recommendation for extensive metabolizers and intermediate metabolizers is 84 milligrams twice a day, but for poor metabolizers it's only 1 pill per day. So you cannot prescribe Serdelga without defining CYP2D6. And that also shows that preemptive testing of pharmacogenetics will be the future to be able to use new effective drugs correctly. Another example is the gene CYP2C19, and very traditional example is Clopidogrel. It's also a prodrug that is metabolized through CYP2C19 to effective antiplatelet substance. But over 30% of patients are poor or intermediate metabolizers, and they cannot get full benefit of Clopidogrel. In Finland, we have about 52,000 patients using Clopidogrel. It means that about 16,000 patients are using the drug without any remarkable effect. And that can be also found out with a very simple gene test. CYP2C19 is also quite important in psychiatric care, as shown here. The same names almost that you had also already in CYP2D6. And it also shows that when you test pharmacogenetic testing, it's better to test the whole profile, not only individual genes. For example, this citalopram or escitalopram, it's relevant in both CYP2D6 and CYP2C19. When we look at different disease groups, this is statistics from Kela, the reimbursement organization in Finland, and there's a number of patients in different disease groups. Cardiovascular diseases, we have carvedilol, clopidogrel, metoprolol, simvastatin, warfarin that are relevant for that patient group, and all of these have relevant genes that could be tested before dosing. For painkillers, I have already dosed codeine, but tramadol, nortriptyline, oxycodone, amitriptyline are also important in this group. And then these psychiatric drugs is only small part of the big list of psychiatric drugs that are metabolized through liver and where CYP2D6 and 2C19 are very relevant. It has been a long discussion about how and when pharmacogenetic testing should be done. Is it preemptive or is it reactive? But if we use the reactive testing, meaning that we have first the complication and then we test the patient, we cannot get the best benefits of the pharmacogenetic testing, because then we know already with that failure that the patient cannot take that drug anymore. But if we have preemptive testing, we can reduce the number of adverse reactions remarkably, and there's a lot of evidence that preemptive testing is more cost saving than the reactive testing. And in different screening and prophylactic studies, it has been shown that if the prevalence of the disease is big enough, then screening is cost effective. And pharmacogenomics, we have quite a big prevalence of polymorphism, and that's why screening, in other words, preemptive testing, is relevant in this context. It's not so sensitive information. In fact, blood groups were invented 100 years ago, and it was the start of personalized medicine. First time we had something that was genetically defined, like blood group, and we started to give blood group related transfusions. Before, we gave the same blood for everybody, and the number of complications was quite high. We are now starting the same big leap to safer medication in pharmacogenomics. In blood groups, we don't ask separately ABO or rhesus. We always get B plus or A plus. It's the same with pharmacogenomics. It's best to just ask the pharmacogenomic screening panel, and then you get all relevant information. But because pharmacogenomics is quite complicated, you cannot print it like A plus or O minus. We have to have tools to collect the all relevant profile to a single device like mobile phone in our application or to a single report in PDF or patient files in clinical practice. So to summarize, pharmacogenomics means fundamental change in healthcare and drug safety. And with that, we can give right medication, right dose, right time to every patient. It's a one-time investment for the patient and valid for the whole life cycle. So there will be new requirements for pharmacists and medical doctors to know the pharmacogenomics. If the patient has, for example, data that they are CYP2D6 small metabolizers, the professionals have to be able to act correctly to give the best available treatment. Thank you for your interest.